Evening. Ben Harris. Ben is the Discovering Reptiles Project Officer at Froglife. And the Discovering Reptiles project aims to improve the participants' knowledge of reptiles and improve the national recording of reptiles throughout the UK. Ben's got a number of years' experience running reptile surveys and ecological talks whilst working as a ranger and project officer in the Lee Valley area of London. So over to you, Ben.
Thank you very much, David. Let me just share my screen. I learned that there was a word actually in German describing the length of time it takes from saying share your screen to actually sharing your screen. Accurately describes it, I feel. But welcome everybody to the Discovering Reptiles webinar. And I feel this follows on really nicely from Kieran's talk a few weeks ago. If you were tuned in then, he was talking about using iRecord for biological records. And my job here tonight really is to convince you that reptiles are a taxon that are really worth your time and your effort to survey, and then to submit your records of those reptiles that you find in your local green space. So just a short introduction to Froglife. So that's the organisation I work for. And we are a national wildlife conservation charity with a particular focus on amphibians and reptiles. And we tend to work in three different ways, in three areas. And we get involved with practical habitat management and creation that benefits amphibians and reptiles. This project in particular really delves into educational workshops and training. And we also do a little bit of research as well. So we're going to kick off with reptile identification. And we're just going to look at what we consider UK native common or widespread species. So this should be an absolute cakewalk for all you botanists, all you entomologists out there that have the knowledge of hundreds of different species. We're just going to focus on 4. But the key thing here is that when you see a reptile, particularly on a visual survey, you're going to get very limited information because you'll catch a glimpse of them and then chances are they will scuttle off into the undergrowth. So we'll be taking a look at what the key features are that you need to be looking out for so that you can get that positive ID on any one of these species. So we'll start with the common or viviparous lizard. And viviparous meaning giving birth to live young. A number of our reptile species here in the UK do that. But the common lizard is a lot smaller than people might think. If you've seen lizards on your travels abroad, in Europe, they're usually a lot bigger. They're only 13 to 14 centimetres. And a good proportion of that is their tail. So they are quite small. And in terms of coloration, they're usually this dull shade of green or brown or bronze. And in the case of newlyborns, they are actually black, and then they grow into their adult coloration. There's usually some form of patterning. So you get these dots and flecks, and they'll usually have a central vertebral line that runs down the back as well. And you can actually use this feature sometimes to determine whether the individual seen is male or female. So we actually have a male here in the top left and a male here in the bottom right. And you can see this vertebral line is quite broken up in places, particularly here in this top left image. In places on its body, it doesn't even look like a line. It just looks like dots and flecks. And that gives it away as a male. Males have more dots and flecks than the females do. The females tend to have this unbroken line down the middle of their body. They will also have darker coloured flanks. And certainly when they're gravid, and gravid just meaning that they are incubating their eggs, they're pregnant essentially, they will appear much bigger than the males. So the later the season gets on, the bigger the females will look. And then finally, if you manage to look at the common lizard for long enough, which is a rare opportunity, You may notice that males have a bulge at the base of their tail, and they develop this bulge during the breeding season. You can see it here reasonably clear, whereas on the female, no bulge at the base of the tail. A very smooth transition from the body into the tail there. Now you're most likely to confuse a lizard with a newt. So you may have noticed this image in the bottom left here is actually a small newt and not a lizard. The best piece of advice I can give you is to approach whatever you've seen, and if it runs off and scuttles off, gives you absolutely no chance of catching it, it's a lizard. Lizards are very fast and skittish, and they're always going to be trying to escape when they can. And a lizard in good health is not going to allow you to pick it up readily. Whereas a newt on land are very slow, very sluggish, and you can basically always manage to pick them up. And then if you do have hold of it, try counting the front toes. Newts actually have 4 front toes, whereas lizards have 5. So that's one way to tell whether you've got a lizard or a newt. But you can also tell from the skin as well. So lizards have scales. You can see all of these images, the scales are very clear. Newts have smooth skin. It's almost velvety when it's dry. When it's wet, if they've just come out of water, it's a bit sleeker. So moving on to the slow worm. So this is actually another one of our our lizard species, but it does superficially resemble a snake in that it doesn't have any legs. But unlike snakes, they can blink, so they can retract their eyelid and they can blink, whereas snakes cannot do this. So if you enter a staring contest with a reptile and you don't know whether it's a snake or a slow worm, if it blinks, then it is a slow worm. But they do grow to be quite large in comparison to the common lizard. They can grow up to 50 centimeters long. And you can use the coloration, actually, of the individual to tell whether it's a male or a female. So we have a male here in the bottom, and it's usually this uniform, dull brown or grey. And then it will sometimes develop this blue fleck or flecking throughout its body. You can see it really clear, very prominently on this individual. Not all males do this, only some of them do. But if you ever see that blue flecking, it's always a male. It's only the males that develop this. And the females, like we have here in the top left, far more bronze in colour, and they have these dark flanks that the males lack. So they're usually quite easy to tell the difference between male and female. The juveniles are very, very small. This time of the year is a really good time of year to find the juveniles. The female can lay up to 25 at once. So the more mature the female and the larger the female, the more likely she is to have a huge number of births. And once she has given birth, they'll quite often sit in numbers of a dozen or more, all underneath the same piece of refugia. And it's a fantastic little find if you do manage to go outside and take a look underneath some stuff. So moving on to the grass snake. One of the other really good ways to tell the difference between whether it's a snake or whether it's a slow worm is actually the distinction between the head and the body. You can see here and here very clearly where the head of the snake is. If you think back to the slow worm images, there's very little distinction. It's really hard to see on a slow worm where the head stops and the body begins. They just look like stumpy little tubes, where it's far more distinct in snakes and certainly in grass snakes. But grass snakes do grow quite a bit larger than slow worms. They're usually between 60 centimeters to a meter long. Sometimes you do come across an absolute whopper of a female that might be 1.5 meters. And it's always something you'll remember the first time you find a snake of that size in this country. It's not something you normally expect to find. You can see across all 3 of these images, they're usually this sort of olive colour or green, dark olive colour. And one of the key features of the grass snake is this yellow collar here on its neck. You see that on all three individuals there. And they tend to have these dark bars running down the length of their flanks as well. Some of you may know that a number of years ago, our subspecies of grass snake was elevated to a full species, and it's absolutely fine to continue calling them grass snake as a common name. Some people have adopted the name barred grass snake, and this is due to the fact they have these bars that run down their flanks, and other species of grass snake across Europe lack these dark bars. You tend to find grass snakes around water. So if you're conducting reptile surveys and you are hoping to find grass snakes, they're usually found within about 400 meters of water, very rarely any further than that. And this is because they are amphibian specialists. They tend to eat frogs and newts, and they are quite active hunters when they are looking to feed, and they will quite often be found in ponds and small lakes swimming about looking for their prey. You can tell the difference between males and females, but it is quite difficult. What you want to do is look at either end. This is actually a female we've got here in the top left. And you can see how the body tapers off very, very suddenly, very quickly. It's very thick set here, and then it gets very skinny round about there. And this is the tail of the grass snake. So they do have, even though it just looks like one big long tail, snakes do have bodies and a distinct tail. Technically, you would have to flip the snake over and identify where the anal gland is to see whether the body stops and the tail begins. But females' tails are usually very short and stubby, and the body tapers off very quickly and suddenly. I believe this is a male here in the right, and you can see there's a much more gradual tapering of the body into the tail here. It's really hard to tell when you're just looking down at a grass snake, a male grass snake, where the body stops and the tail begins. So that's one way you can try and identify male from female. And the other way is actually again to look at where the head meets the body, and that's really distinct on female snakes. The head fits onto the body with this very distinct meeting. And the males, it's far less distinct, far smoother. They have much skinnier heads than the female. But this won't always work. It's very hard to sex grass snakes if you just catch a glimpse of them. And then finally, our last common or widespread species, and certainly not as common as they used to. We are losing adders at a very rapid rate, but they are still considered widespread. So they're a very successful species in terms of their range. And you do get them in the far reaches of Scotland, all the way down to southern England. And you don't get grass snakes very often in Scotland at all. In fact, in terms of distribution, you tend to only find grass snakes in southern England and particularly the east of England as well. So adders are widespread, but not as common. And they are a little bit smaller than the grass snake. So only between 60 and 80 centimeters, but they're far stockier. That's the one thing you'll notice when you see an adder basking, is they're a very thick set snake. This is probably why they're so successful in terms of their range, because it allows them to survive in much colder conditions than our grass snakes. In addition to that, they do incubate their young inside of them as well, whereas grass snakes are one of only 2 of our native terrestrial egg-laying species. So the key identifying criteria, that key feature of the adder, is that zigzag pattern that runs down the length of the body. You can see it across all 3 of the images here. And it usually starts with a V on top of the head, continues down the length of the body. I've never seen an adder without that zigzag pattern, but I've heard that they do exist, and the zigzag pattern can be quite variable, but there's usually some sort of pattern that resembles a zigzag shape that runs the length of the body. And in males, this is always black. So we have a male here on the right hand side, and in females this is usually some shade of dark brown, but can also be black. So if you see an adder with a dark brown zigzag pattern, it's always going to be a female, whereas a black one is usually a male, but can sometimes be female. And there is a difference in coloration between the two sexes as well. The males do tend to be this sort of greyish colour. Females tend to be a more golden brown, dark brown colour. This is a juvenile we have here in the top image, and they tend to be that gingery brick red colour. Really similar colours between the body and the zigzag pattern, but you can still see that zigzag pattern there. It's usually a shade darker. And you can actually tell the difference between grass snakes and adder by taking a look at their eye. Adders have this amazingly menacing stare. They have this red iris with a vertical pupil that almost screams, "Don't come any nearer. I am venomous. You know, you don't want to tangle with me." Whereas grass snakes, if you can think back to that image, they actually have they've got a circular pupil with a yellow iris, which is far more inviting. But again, you may not want to pick up a grass snake, as they do release this kind of, people call it foul smelling, but it's just this musk that I wouldn't consider foul, but certainly distasteful if you don't want to get it on you. Okay, so let's just briefly take a look at the general life cycle of a reptile here in the UK. And this does differ a little bit based on the species, but also the annual weather for that year, as well as the location as well. So sites further south are going to have reptiles leaving hibernation earlier, and likely are going to have reptiles more active later in the year as well. But generally speaking, between February and April, the individuals are going to be leaving their sites of hibernation, or their overwintering sites. Adders tend to be the first species to emerge, followed by common lizards, slow worm, and then grass snakes probably a little bit later. And then between the months of March and May, most of the individuals are going to be looked to be breeding, particularly the males, whereas the females may not breed every single year. But the strategies are very different across all the different species. The lizards tend to be a bit more combative, whereas the grass snakes will create this mating ball with many females trying to access one female. And then you may have heard of, and certainly I've never seen it, it's a very rare sighting, the dance of adders, where you get a very gentlemanly-like battle between two adders that, no biting is allowed, considering they're venomous, where they actually use their bodies to try and force one another to the ground, and then the winner gains access to that space, which hopefully should have a female in it. And then between the months of April and August, so the majority of summer, all of the species that I've just outlined, other than the grass snake, will incubate their young inside of them. So they will be finding the best basking spots to ensure that the eggs inside them will incubate optimally. And then the grass snakes will be looking for potential egg-laying sites to deposit their eggs. You don't get as much activity during the summer in reptiles than you do at the start of the season, so between February and April, and at the end of the season, because the majority of the individuals have done exactly what they want to do. They'll have fed, they would have finished their breeding routines, and it's much easier to get to their preferred temperature range. So July and August in particular are actually poor months for surveying for reptiles, which is sort of counterintuitive when you think, well, reptiles are, well, cold-blooded, people call them, and they're looking to get as warm as possible. We'll cover that in a later slide, but yeah, you do see less of them in the summer months. And round about now, so September and October, you get this flurry of activity, this second peak of activity, where the adults, and as well as the newly born young, are looking to feed as much as possible in order to put on that weight, put on the calories to survive that overwintering period, which tends to be between October and March. So we've had a cold snap recently, certainly where I am, And this is really going to start triggering many of the species, many of the individuals to start looking to begin their hibernation cycles. And it's worth noting here that they will use the same hibernaculum year after year. They're very site loyal. So once they've found somewhere they overwinter, they will continue to use that site. And it's really important that site managers identify if they have these sites on their land and do all they can really to ensure that they're not dismantled and preferably protected from any disturbance, so grazing or too much public disturbance, particularly dog walkers, because reptiles very, very loyal to the sites they hibernate in. Okay, so we're going to go over a few of the sort of key pointers on how to go out and do your own reptile visual survey. So what you need to do, why it's good, and how to do it. Now, before we dive into the meat of that, excuse me, it's worth considering how reptiles manage their body temperature, because in understanding that, it allows us to exploit their behavior so that we can get the most out of our surveys. A thermoregulation is just the process by which any species manages their temperature within a certain range. So I do it, you do it, most species do it, but we do it endothermically. We're endotherms. Reptiles are ectothermic, so they will use their environment to thermoregulate. They don't have these internal processes such as sweating that allows them to manage their temperature. It's a bit of a misnomer that we actually call them cold-blooded, because they're still looking to reach a preferred temperature range of between 25 to 30 centigrade. They just do it very differently to how we do it. So they will use their environment to cool down if they're too hot, just as much as they will use their environment to heat up as well. And the majority of the reptiles in this country are what we call heliothermic, and they take their temperature directly from the sunshine. And we call this basking. That's the behavior in order to get to that preferred temperature range. Slow worms are kind of the exception. They will bask, but they don't like to do it very often. They are thigmothermic, and they will take most of their temperature from conduction of materials within their environments. They're usually found underneath things rather than on top of things or out in the open basking. Adders and common lizards are particularly efficient at thermoregulation. They're really good at flattening their bodies against things. So not only do they increase their surface area for the sun to hit their body, but they're also increasing the surface area to conduct heat from items that retain heat well or that heat up quickly in the morning sun. And that's reflected in distribution maps as well. So adders and common lizards, probably the two most common reptile that you'll find in Scotland, where temperatures are far cooler than they are in particularly southern England. So when is the best time to survey? So we've covered the general life cycle of the reptile. So it's no good surveying when they're hibernating. So between October and February is not going to yield you many results. Though interestingly, it's not a technicality I normally care to correct people on, but reptiles, they brumate rather than hibernate. And you do get these small peaks of activity during winter if it's warm enough that you wouldn't otherwise get in what we would consider hibernation. So it's not impossible that on a warm day in December, a reptile might crawl out of their hibernation site and bask for an hour or two, or maybe even feed for a little bit. So they are active between February and October, and you will get results during any of those months really. But the best time to do it, if you want to optimize your results, are between March and May, when they're coming out of hibernation. They're looking to bask a lot, and they're looking to breed, so there's a lot of activity. But also in September and October, when they're looking to feed as much as possible before they go into hibernation. And the time of the day is important as well. So the morning is good. because in the morning they are looking to heat up to their preferred temperature range after a cold night. But usually when the sun is at its peak, it's going to be warm enough that they don't need to be basked. They don't need to bask. They don't need to be in the open. And particularly in the summer months, they'll be able to just take their temperature from the ambience of the day rather than having to bask in direct sunlight, which makes it difficult to see them. But when the temperature drops later on in the evening, you may find there's more activity in reptiles as well. And you really have to tailor this depending on when you're doing it and the type of weather that you're experiencing as well. So the best temperature range is anywhere between sort of 9 and 18 C. But in cooler temperatures, so if you think about doing a survey in March, you're looking for those cool, crisp days where the reptile is going to be out probably later on in the day and for longer. Whereas in the summer months, it's going to be out earlier and it's probably not going to be out for as long as well. The warmer the day in the summer, the more cloudy it is, the better. So if you think of the sort of temperatures we get in sort of June, July and August quite often exceed 18 centigrade. It's why you don't see reptiles very often, because they just don't need to bask. Once they're at that preferred temperature range, they just won't be out basking. So that sort of hazy weather where they're struggling to get to that optimal temperature range is best. And any rain or really strong winds make it sort of unsuitable for surveying for reptiles. In rain, they're just going to find cover and you won't find them. But I guess the exceptions to that rule are sort of April showers. If you think of a quick bout of rain, so an hour or so of hard, fast rain, it's going to cool them down very quickly. But when that sun comes out immediately after, it's going to encourage them to go out and bask and raise their temperature up again. So that's sort of one exception to the rule in terms of rain isn't good for surveying. So in terms of survey method, why is a visual encounter good? Or why is a transect good for surveying for reptiles? I would argue it's the hardest one, but it's also the lowest effort. And it just makes it so fantastically accessible. If I wanted to go to a site tomorrow morning that I've never been to before, and I wanted to do a visual encounter survey for reptiles, I can do so. I just need to go. I don't need any equipment. I don't need any permission from the landowner, as long as there's public access. And it's super low effort. It's just another word for taking a jolly in your local green space, really. And it's that accessibility to surveying which makes it a really good one. But it's not a great scientific method. It's very inappropriate at determining absence. So if you see a grass snake on a site, you can confirm presence 100%. However, not seeing a species, you'll never be able to determine absence. And this is really pronounced in reptiles because, one, they're cryptic, so they don't like to be seen. And two, some reptiles have very low occupancy in habitat that's suitable for them, mostly because they really struggle to colonize areas that are suitable for them, and because we've fragmented the landscape that they live in. But a visual survey is really, really poor at determining someone saying, well, there's definitely no reptiles there, because it might be that you just didn't see reptiles the 8 or 9 times that you went out to do your visual survey. And maybe you were unlucky, but you can't determine absence from a visual encounter. And then on top of that, the survey results really aren't comparable between surveyors because there's no standardization of survey effort. You're going to get very mixed results if you have 2 surveyors on 2 different sites because of the different level they're at. Usually, one surveyor isn't comparable to another. One might be a beginner, one might be more experienced. And even over time, the same surveyor is going to get better at spotting reptiles and understanding where the reptiles are likely to be. So even though you might see an increase of numbers on a site, it may not reflect the fact that reptiles are actually doing well on that site. Okay, so how do you do it? How do you do a visual encounter? We know how to identify the 4 common and widespread species. We know a good local green space where we want to do some surveying, either because we know they're there, or even better if we don't know they're there, but there's the potential. How do we do it? It really is as simple as taking a walk. And the real skill in this is understanding where you need to walk and where you need to look. We will cover that in the next few slides. But you do just walk relatively slowly compared to your normal walking place. I would say keep off well-trodden paths. So if you're on a site that has a lot of footfall, try and find the paths that are least disturbed. Reptiles do not like disturbance. If they are continuously disturbed, they will probably move on to another area. And you do just need to sort of tread lightly, avoid casting a shadow in front of you, as not only will it scare them off, it's really difficult to see into your own shadow on a bright day when you've got the sun shining down on top of you. And patience is a real virtue when it comes to reptile surveys. You just might not see them the first time, the second time, the third time. And you just need to be persistent and patient as to when you might see them. And you will build up, if you do have reptiles on a site, you'll build up a knowledge of where's the best place to see them. We call these reptile hotspots usually. And it's a fantastic feeling when you've discovered reptiles in an area that you didn't know they were there, and then you begin to learn exactly where they like to hang out. It's a very rewarding experience. But you can look out for dump refugia. They will bask on top of and underneath discarded plastic sheeting, road signs, tyres, any old piece of rubbish that heats up quicker than its surrounding environment. And take a look underneath it. Taking a look underneath it is more likely to yield you results in terms of slow worms than anything else. And I will show you a picture of mosaic basking in a second or two. But one other top tip is if you do hear a rustle and you think it could have been something, take a few steps back, wait 5 minutes, and lizards are particularly prone to this. They will return to the same spot, exactly the same spot. They're creatures of habit, and then you'll be able to tell whether it was a lizard or not. They do this so often, it's something you can take advantage of. So I'm going to highlight some of the best habitats to focus on in your local areas. I'm not going to go through all of these, but reptiles can be anywhere, and that's because we have just fragmented their habitat, and a population may just be clinging on to that particular site because it's the last site that they actually can move around. It doesn't need to be ideal. But that being said, heathland is fantastic. If you have an area of heathland near you that is under-recorded, which isn't often the case because normally heathland is very well recorded for reptiles, heathland provides everything a reptile might ever need in its life. Great areas for hibernation, great areas for feeding, great areas for basking and cover. But barring heathland, any of these sort of suggestions are a good place to start. And what you're really looking for are sites that have really diverse vegetative structure. Areas of bare ground, particularly if they're south-facing. Areas that have short grass transitioning into medium or long grass, and then areas with scrub, and then sort of woodland edge or regenerative woodland. Really good places to start. Even better if it's a mosaic. So if you have patches of each of those things that I've just said littered throughout the site. You kind of need to avoid anywhere that has a sort of homogeneous sort of structure of vegetation. So where to look? So you know where you want to go, you know how to identify them, where do you want to be looking? So on your transect, where do you train your gaze? If you're going to take away anything, it's this. Look in between different types of habitat. So if I was here in this image and this looks like heathland, I wouldn't be looking in the center of this grass here. I wouldn't be looking in the center of the heather either. look at where the heather meets the grassland. So it's called an ecotone, where two different types of habitat transition into each other. And that is where you will find the majority of reptiles, usually basking. It doesn't matter if it's woodland edge into scrub or scrub into shorter grassland. It's those habitat interfaces that you really need to be looking at, and try and find a path. And paths are really good for this usually, because paths are usually short grass that transition into short grass and hedgerow. That's where you're going to find the reptiles. So here in this image, if I was there, I'd be walking this path here, looking at where the heather meets that path. Wouldn't be venturing into the middle of the heather, you know, where it's thickest. I'd be looking at the edges where it meets the path. I may even venture up here to where these trees meet the heather as well. And perhaps you might find, if it's a particularly warm day, reptiles there as well. So yeah, just reinforcing that areas with varied height structures are really good. And you want to be looking at where those varying height structures meet the edges of habitat. So old stone walls are superb. They are the best areas for hibernating reptiles. And certainly earlier on in the season, taking a walk along the edge of a stone wall and seeing if there's any basking reptiles is a good place to start. And any areas that are close to shelter and south facing. So the aspect is usually quite important. If you have a bank that is south facing and has short vegetation on, if there are reptiles on their site, I would bet that's exactly where they would be. That would be a perfect reptile hotspot. You can see in this image, really sunny here. We've got this sort of scrubby patch here, this shorter grassland. You'd be looking along here at the edge. That's where you want to be training your gaze. I said I'll show you mosaic basking. This is exactly what it is. So we've got an adder here. You can tell from the zigzag pattern. And if you weren't looking out, you'd miss it completely. A lot of reptiles will just expose part of their body. I'm assuming they do this to protect themselves from predators, but a lot of them will just mosaic bask like this. And this is a picture perfect basking position. So the habitat interface I was talking about, we've got two adult adders here. You can see this thick vegetation at the back and the shorter grass here. And it's just so that when they are disturbed, they have somewhere to escape to. They're going to be slithering back into that long vegetation where you can't pursue them should you disturb them. But they like to bask in open sun. That's exactly what they're doing here. And again, even a log pile, you can sort of see the interface here between where the reptile wants to bask and where it's going to escape to should it be disturbed. So that's what you should be thinking, like, where is this reptile going to escape if I see it? If there are no places for it to escape, then it's unlikely to be basking there. It likes places that it could run off to and not get caught and eaten. And if you are around any ponds or wetland areas, do keep your eyes out. In the middle of these places, grass snakes have been known, and we quite often get videos sent in from the public of grass snakes taking a swim. Presumably, they're either looking to feed, so they're looking for frogs or newts, even small fish sometimes, but it might be a male looking for a female earlier on in the season as well. They're very, very happy in water. It does not deter them whatsoever. Do keep your eyes peeled if you are near any good ponds. And when you do sight a reptile, we would recommend that you use the Dragon Finder app. So I guess this is what we use for our amphibian and reptile biological records. You can use it to identify things. So it will ask you a set of questions so you can correctly identify it if you couldn't quite remember how to identify things down to a species level. And you can submit the data to. By submitting it directly to us, it really does help us inform exactly where we need to target our habitat creation, our habitat management, but it also helps us put together projects that require funding as well. This data we submit to the NBN on an annual basis. So we don't just hold this data, it does get submitted to where it's going to be most useful. And any local record centre can request that data from us, and we will share it openly with them. But please do use Dragon Finder to send in your sightings. And this is what this presentation is all about, really. We need your help. It's a little bit late now to start, but hopefully in the new year, if you know of a green space next to you that might be particularly under-recorded, and there is a real paucity of data when it comes to reptiles, please do carry out as many as you can. We recommend 2 to 4 visual surveys and submit the records to us. So using the Dragon Finder app, areas that are secluded and that contain that suitable habitat. So anything that has that diverse vegetation structure I was talking about. Okay, so hopefully you've forgotten everything I taught you in the first half of the presentation, and I'm going to test you very quickly on some of the species we learned about. So I want you to try and identify the species. Okay, and if you can, and you won't be able to do this for all of them, try and identify the sex of the species as well. Okay, so I think Kieran has control of the poll. So when the image pops up and give you about 20 to 30 seconds, you'll be able to vote on what you think it is, and then I will reveal at the end what it was. Okay, so this is our first one. Excellent, right on cue.
Don't be shy, guys. It's anonymous. We don't know who is guessing what. And this will be a nice test, not just of your reptile knowledge, but of how good Ben is as a teacher as well. So, we won't just judge you for getting it wrong. We'll judge Ben too. So please do have a go. It should have popped up.
Yeah, and it's completely anonymous as well, so that there's no fear of getting it wrong. So I'm going to end the polling here, and I'll just share it. And this is exactly what I wanted to see. So you're all getting that this is an adder. Great stuff. So I'm assuming most of you identified that zigzag pattern that runs down the length of the body, that gives it away as an adder. And I would agree with the majority here that this is a male as well. A black zigzag pattern, the sort of grey, dull colour. I would probably classify this as a male, but you know, without further examination, it could be a female, but I would tend to go with male there. So well done to the 98% of you that got adder. That's a fantastic result. Let's move on to the second one. Okay. Okay, so the poll's up. Make your votes. Okay, fantastic. So the majority of you have got common lizard, which is correct. So you can see that it's got scales, which the newts lack. You can't count its toe. You can see its toes, but you can't count all of its toes. But it's the scales that really identify it as a common lizard rather than a small newt. The coloration to some extent as well. You can see that it's got some lines and some dots. It's not quite as uniform as small newts are in its coloration. In terms of its sex, it's really difficult to tell this one. It does have a distinctive darker flank than it does on the coloration at the top of its body here. But I can't see its vertebral line, so I can't make a judgment there. I can't see the base of its tail, so I can't make a judgment there. And it's hard to tell whether this individual is basically pregnant or not, or gravid. So I would probably classify this as I don't know, not enough information. But if someone forced me to make a choice, I would say probably maybe female, based on the fact that it has darker flanks. But I would happily take an expert's opinion on whether this was male or female. Okay, so image number 3. Great stuff. Thank you for your votes. Let me just share them. There we go. Great stuff. Most of you got this as slow worm. Bit more difficult than some of the previous ones we've looked at. You can't see its head, which is a real giveaway when we look at the distinction between the head and the body. With slow worm, there's very little distinction. But you can tell it's slow worm based on its coloration. You just tend not to get that coloration in snakes. It doesn't have a zigzag pattern. And this is a female. So you can just about see the dark flanks here. Just about see the dark flanks here. And I don't know if any of you can see there just about a vertebral line that starts on its head and sort of runs down the length of the body, but in a very broken line, giving it away as a bit of a female. This is a female slow worm. So well done to those that got it right. Number 4. I think we've got 8 of these in total, so this going to try and get through these a little bit quicker than the previous ones. As I know, if we're planning to finish at 7:30, it won't give us much time for questions. Okie dokie, here we go. So you're all smashing it. I don't think I've seen anything less than 97% correct. And you're right, this is a small newt. So small newt is the name we actually give if we don't know the difference between as to whether it's a palmate newt or a smooth newt. They are small in comparison to great crested newts. So this could be either a smooth newt or a palmate newt. But it's certainly not a lizard, which is exactly what I was looking for. So fantastic stuff here. Yeah, and I would say I've not enough information here. I would not hazard to guess as whether this is a male or female newt. Moving on to image number 5. Yep. So most of you have got adder here. A few of you said grass snake. So you can still see that zigzag pattern that gives it away as an adder, which is what we're looking for that positive ID. And then the majority of you also got female, which is really great to see. It's got that brown zigzag pattern. It's not black. If it was male, it would be a black zigzag pattern. So we can identify this as female. So female adder is correct. Okay, number 6 of 8. Great stuff. Most of you got slow worm, top notch, and it is a male. So some of you may have seen a few blue flecks here, gives it away as a male, but it really highlights how it is very variable on males as to whether they develop a lot of it, very few of it, or not at all. It is a slow worm, no distinction really between the head and the neck, and as blue flecks give it away as a male. So this is the penultimate one. Okie doke. Great to see most of you got this as a grass snake. You can see the dark bars here running down its body. Just about see that yellow collar, which is what you're looking for when you just see a flash of something in the undergrowth. And I don't know whether this is a male or female. I can't see its tail, so I can't make a judgment there. Can't really see its head, so I can't make a judgment there. If I was forced into an answer, I would probably say female because it looks to be a little bit larger than the average grass snake, but it might just be an older male, and they do grow to a good size, the males, if they're given the chance to do so. But I would say there's not enough information there, which most of you put, which is great. And then finally, this is the last one. Could we get the polyp for this one? Okay, cast your votes. Let's see if we can get 98% correct for the final one. Or 100%. 98% far too low. Need to shoot far higher. We did it at 98%. Great stuff. So this is a common lizard. You can see it's got scales. You maybe can just about count the front toes, but you can see it's scaly all over, which really gives it away as a lizard and not a newt. And I would probably agree with the majority of you here that this is female. Tends to, it doesn't have a bulge at the base of the tail. It's got this quite prominent vertebral line, not too many dots. You can maybe see a bit of a dark flank there in comparison, just underneath the sort of dorsolateral line. So I would hazard a guess this is female. So well done to everybody on that. So fantastic to see so many of you know how to identify reptiles. There was 90% of you getting it right every time there, which means you can do it. You can go out, you can go and do your surveys, and it's going to be much harder than seeing a picture for 30 seconds, but please do go out and submit your sightings. I'll show you an image just after this, after I say thank you to my funders, on the number of records we have just for the London area. You might be surprised at how low it is. But this project is made possible by the National Lottery Heritage Fund. So thank you to those guys for funding such a fantastic project. I will take your questions. And I'll bring this screen up after I've shown you this, so you can email me if you have to dash off but want to email me any questions. If you can see this map here, it might be a bit small if you're looking on a small device. These are all the reptile records the NBN holds within the sort of London area. So we've got 9 different species, but some of these records stretch back to the early 1900s. We've only got 100 common lizard, 23 grass snakes, 32 slow worms, and I know for a fact that they're certainly within this area of London, and we don't have enough data on where reptiles are and how they're doing as a species. So please do go out and send us your records. It is really important. And it's volunteers such as yourselves doing so that make the difference here. So thank you everybody for joining this evening. It's been a real pleasure to deliver this presentation to you. So I am going to take some questions now, I think.
Yes, thanks Ben. We are quite on our time, so we're going to have to stick to just a couple of questions. I'll ask Maria to pull a couple out of the forum. So Maria?
Yeah. There's been loads of interest, and I'm really just going to have to pick a couple of things out. There's been some people interested in habitat management, with best ways of managing habitats. James Orr said that, and also a bit about whether grouse moors and that kind of habitat. That was from Lee Stewart. Is that quite a good habitat for reptiles or not?
Grouse moors?
Yeah.
So heathland is good. So so areas where they have heather is good. Areas that are managed for particularly driven grouse shooting is bad, because they regularly burn areas that will just decimate the population of reptiles on those areas. The controlled burning of heathland for grouse shooting is no different to the heathland fires you've seen in the south that are caused by barbecues. They are still just as destructive for reptile populations. So I would argue that the burning of heathland is a poor management practice for reptiles, if that's your focal species. But heathland in general is if it's allowed to develop and is managed sympathetically for reptiles, is the best habitat that we have for reptiles in this country.
Yeah, that's yeah, thank you. And then just something, one more. So last thing, Wendy Knight's quite interesting question about whether there's any variation of gender with temperature. Is it, you know, are they kind of one of those groups of animals where there's some effect of temperature on whether they develop into male or female?
It would be something I would have to look up, actually. I know for frog spawn, that's it's just a couple degrees temperature that can determine whether it develops into a male or female. And I would hazard a guess that yes, that the difference in temperature as it develops determines whether it's a male or female. But I wouldn't be sure. I wouldn't be able to answer with certainty for that.
Okay. Thanks, Ben. I think that's going to be about all the time we've got for questions, I'm afraid. But Ben very generously provided his email address, so you can all bother him personally, or do come along to the LNHS's Facebook forum and continue the discussion there. So thanks once again to Ben, and thanks to everyone for coming, for all of your questions, for all of your interest, and for all of your learning to be really, really good at identifying reptiles. Our next virtual talk is going to be on the 15th of October, and it'll be More Than Weeds, the Flora of London's Pavements, and be by Sophie Le Guil. See our website for details. And